Hello everyone and welcome back. Today we will be covering some of the main ideas from chapter 4 that will look at cells, various types of cells, and components of basic cell structure. Again, just to reiterate, always utilize your textbook as your primary study source, and then use these PowerPoint slides to solidify your learning. So let's get started. All life begins at the cellular level, so therefore it is going to be essential for us to learn what a cell is and how they function. Back in chapter 1, we looked at 7 characteristics of life, or 7 things, components, or processes inherent to all living systems. And the cell is a unit of life that displays all 7 of those characteristics. So for something to be considered alive, now again, we're not referring to gray areas like viruses here, because those are subcellular entities. But for us in this class especially, when we're talking about life, it has to at least be one cell. So things like bacteria and protists are one-celled life forms, very tiny, all the way up to huge multi-celled life forms like plants and animals, and also us. Think of a cell as a complex machine that is capable of supporting life processes. So very intricately designed, and we will explore some of those complexities throughout the semester, and we'll start that here in chapter 4. When we think of cells, we typically think of microscopes. So by and large, cells are very small and we require a microscope to see them. So we're looking at somewhere about the micrometer scale. That's what that little symbol stands for. It's a micrometer. Very tiny, obviously, but not all cells are microscopic in size. So we have things like chicken eggs, ostrich eggs, or even frog eggs that are one cell, and we can see them with the naked eye. So cell diversity is very complex. We could spend an entire semester on cell diversity itself, but to orient us to the relative size, we are going to be concerned with this area here. So we'll be looking at different types of cells, like animal or plant cells, and then components of cells. And as we can see, we have living organisms like bacteria that are actually the same size as some organelles that we have in our cells. So again, this is just comparing and contrasting relative size of various cells to one another. There's something called the cell theory, and there are various components to the cell theory, but by and large, all scientists will adhere to this cell theory. So make sure you are able to recognize these components of the cell theory. So it states that all cells come from pre-existing cells and that the cell is the smallest unit of life. So really grounds us into this framework with which we will view biology for the rest of the semester. Now, according to this cell theory, cells do not spontaneously arise. Again, they come from a pre-existing cell, and this theory may contradict some worldviews. So some fundamental questions that may arise when looking at this may be like, well, where did the first cell come from? And that is outside the scope of this course, and in my opinion, biology in and of itself. But that question will ultimately depend on your worldview. So was the first cell created or did it evolve randomly? But from there, okay, we definitely adhere to this, that every other cell from that first cell came from a pre-existing one. Cell diversity. Again, I mentioned that cells are extremely diverse in type and size and organization. But for this chapter, we will be looking at two categories of cells. And we will start looking at the science of classification, and we call that taxonomy. And that is this chart over here. You may be familiar with the taxonomic hierarchy of life. You may have learned this in high school. Kingdom, phylum, class, order, family, genus, species, with a species being the most specific niche of an organism and a domain being the largest category, and it's the highest level of life. So if we were to establish something as alive, so we've gone through all 7 of those characteristics and it met all 7, we can say, okay, this is a life form. Our next question would be, what kind of life is this? Is it a bacteria? Is it an archaea or a eukaryote? And that would be starting our taxonomy, and we start at the domain level. Bacteria and archaea are both prokaryotic cells, meaning their cells are smaller and do not have membrane-bound organelles like a nucleus. I don't want to say that they are simpler life forms because life is never simple, but there's less stuff in there compared to eukaryotic cells. These are cells that are much larger and have membrane-bound organelles like the nucleus. So think of plants, animals, fungi, etc. So be familiar with the 3 domains of life and what kind of cells they are. So domain bacteria is a prokaryote. Domain archaea are also prokaryotes, meaning no nucleus. And then domain eukarya have nuclei in their cells. And we are eukaryotes because our cells have nuclei. As we explore the differences between prokaryotic cells and eukaryotic cells, and later in the semester we will spend a week on life forms themselves. So we spend a week on bacteria, we'll spend a week on plants, we'll spend some time with fungi, etc. It's important to remember that while there are many differences, there are 4 unifying features all cells have to have. So again, think back to those 7 characteristics of life. For something to be alive, classically, it begins at the cell level. And what does it mean to be a cell? For something to be a cell, it has to have these 4 components. Okay, so at the very least, all cells will have a cell membrane. So this is a fluid, flexible skin, you could think of, surrounding the internal components of the cell. It lets things in and out, it holds the cell together, etc. All cells have a cytosol, or cytoplasm, you may have heard it called. This is analogous to like the blood of the cell. It's just an internal fluid that suspends the organelles, allows for movement and transport, fills out the cell so again that it is able to hold its shape. All cells have ribosomes, and this is a type of organelle that we'll discuss later. But these are the tiny little dots that you often see inside of a cell, and they are where proteins are made. So a DNA message will be converted into RNA, and then the ribosomes will read that RNA message and make a protein. And lastly, every cell has to have some genetic material in the form of chromosomes. So chromosomes are strands of DNA that are wrapped around proteins, and all cells will have DNA genetic material with some protein in there, and we call that combination of DNA and protein a chromosome. So definitely know these four characteristics, ubiquitous to all cells. Detailed version of this picture cell. And if we look at this picture, we have an animal cell and a plant cell side by side. There are some things that the plant cell has that the animal cell doesn't have, and vice versa. I just want to point that out really quickly. The plant cell has a cell wall, which is a rigid structure surrounding the outside of the cell. It gives it its shape, aids in some protection. Animal cells do not have cell walls, so that's one thing the plant cell has. Animal cells do not. Plant cells also have chloroplasts, which is an endomembrane organelle responsible for photosynthesis. And plant cells also have a very large central vacuole or vesicle, which can absorb a lot of water to make the cell more turgid, and at the same time it can expel water if the plant needs water. It just has acts as a reservoir there. Animal cells, by contrast, have a feature called a lysosome that plant cells do not. And you can think of these as little vacuum cleaners of the cell, so they will absorb waste and break it down using lysolic acid, or action, excuse me. The same mechanism that Lysol that you would spray on a table would use. The chemicals in Lysol essentially burst apart bacteria and virus components, and those same chemicals are found in the lysosomes of animal cells. Everything else, though, both plant and animal cells will have. So again, the nucleus is this membrane-bound house or container for the chromosomes, or in this case, chromatin. Chromatin is just a more diffuse, stringy version of a chromosome. And then we have an extremely compact, dense component where the chromosome is tightly coiled, and we just call that the nucleolus. So various components to a nucleus that we can see. We have the outer membrane of a nucleus called the envelope, the chromatin, which is all of the DNA and the protein action that will eventually form chromosomes. and then a very, very compact, darker little spot within called the nucleolus. We have other vacuum cleaner type structures called peroxisomes, which again metabolizes waste, act as vacuum cleaners. They will sort of wander and break down substances not needed. We have another component of our endomembrane system, the endoplasmic reticulum. Two versions of that, rough and smooth. The rough ER, as we can see, comes right off of the nucleus, and it is studded with tiny little dots, and those tiny little dots are the ribosomes. So that's one way you can differentiate the two types of ER. Rough is directly attached to the nucleus, studded with ribosomes, and then smooth, not directly attached, and of course no ribosomes, giving it a smoother appearance. And then the functions for those two, the rough ER makes proteins, again, studded with ribosomes. That's the job of the ribosome, to synthesize a protein, and the smooth ER will synthesize lipids or fats that we looked at in chapter 3. Because animal cells don't have a cell wall to give them structure, animal cells have an internal skeleton called a cytoskeleton made up of microtubules and centrosomes. So these are tiny little structures that can essentially build up cytoskeleton fibers, sort of originating like this. Various filaments that make up the cytoskeleton, again just giving the cell its structure. Plasma membrane, lysosomes that I went over earlier. One more component of that endomembrane system called the Golgi apparatus, and this just will further modify proteins, and it also packages things and can send them out of the cell. Cytoplasm and mitochondria. Mitochondria are the powerhouse of the cell. You've probably heard that at some point. And this is where sugar is broken down into usable energy for the cell. So again, as you're reading, make sure you study all of these structures and know their function. So be able to label these types of cells and then perhaps match the structure with its function on a quiz or exam. So we can go through and look at these structures a little bit more in depth. So we can start with the plasma membrane or cell membrane. Those terms are interchangeable. Again, think of this as the skin of the cell, and it allows things in and out. But some things can move freely in and out of the cell. Other things have to be allowed in or out. That's what it means to be selectively permeable. So things like water, okay, can move freely in and out of the cell, but other things like sugars, fats, hormones, etc., have to be allowed in. And if we look at a model here of a plasma membrane, we can see there are a lot of components to the plasma membrane, and actually it is made up of 3 of the 4 types of macromolecule that we learned about in chapter 3. So the majority of all plasma membrane are made of this phospholipid bilayer. So looks similar to a triglyceride, if you remember the structure of a dietary fat, but instead of 3 tails like a dietary fat has, this only has 2. So a phospholipid makes up the majority of the cell membrane, and we can see that is actually a bilayer, or there are 2 layers of this phospholipid, and they will orient like this because this head is able to dissolve or be in contact with water, while these tails are not. So that is essentially what creates the skin of the cell. Then we have carbohydrates that make up the component, a component of the cell membrane. We have things like cholesterol. Again, this was a type of lipid, a steroid that we learned about in chapter 3, and this just helps keep things fluid. And we then have proteins. So these proteins are actually what allow certain things to come in and out. And because we have all these various components working together, we call the plasma membrane a fluid mosaic model. If you are a plant cell, just outside of the plasma membrane, you have your cell wall. And unlike the cell walls in prokaryotes, plant cell walls are made of a different type of complex carbohydrate called cellulose. So definitely be able to tell me what kind of cell has peptidoglycan as its cell wall, what kind of cell has cellulose as its cell wall. And again, the function of a cell wall is to provide structure, give plant cells a square shape, and to keep the plant cell from bursting if it were to take on too much water. Animal cells, like I mentioned, don't have a cell wall, but we have an internal cell skeleton and also a sticky fibrous network outside called the extracellular matrix. So it's really a scaffolding that helps to keep cells in position relative to one another, and it's made of the protein collagen. So a good example of this scaffolding function of collagen is that as we age, the collagen, or that scaffolding that holds our cells taut, begins to relax and get loose, just a consequence of age. So especially on our faces as we age, those collagen fibers, which is a type of protein, will relax and we can see that wrinkles or divots form because that scaffolding effect of our extracellular matrix is not as taut as it was when we were young. The nucleus. So this is the genetic control of the cell, the command center, if you will. So nucleus for eukaryotic cells, and again, the nucleoid we find in prokaryotes, the control center of the cell. Now, if we're going to just talk about the nucleus here, it's a double membrane bound organelle, so it's part of that endomembrane system. And again, it has pores that can allow certain things to come in and out. The nucleus is home to the genetic material. So if we're talking about prokaryotes, eukaryotes, but again, eukaryotes only have nuclei, houses the chromosomes. And on chromosomes, we have genes. And the definition of a gene is the instruction for how to make a protein. And as I mentioned earlier, DNA. So here we have naked DNA, a double helix. This was one of our macromolecules, a nucleic acid that we learned about in chapter 3. Naked DNA will wrap itself around little proteins called histones, and this just helps to package the DNA later. And it is this protein DNA complex that we call chromatin. Chromatin will continue to wrap and coil and condense into structures called chromosomes. So we have 3 different levels of packaging here. We have naked DNA that will wrap around proteins to make chromatin, and that chromatin will then condense and coil up into a structure called a chromosome. So when a protein needs to be made, and we will learn this process later in the semester, but basically DNA, a gene on a strand of DNA is read, and an intermediate messenger called RNA is created. And then the RNA will exit the nucleus and find a ribosome, and that ribosome will read the message on that RNA and make a protein from that. So Ribosomes are the site of protein synthesis. We find them in the cytoplasm and also embedded in our rough ER. And again, this process, okay, we will get into in the coming weeks. Looking at the endomembrane system a little closer, so again, endomembrane, inner skin, or these are structures that prokaryotic cells do not have. And you can think of it as a continuous network of membranes that just do different things for the cells. It adds in the complexity of a eukaryotic cell. And these are the structures found in the endomembrane system. And some of these we've already gone over a little bit. But we have our nucleus, the endoplasmic reticulum, the Golgi apparatus, lysosomes, and vacuoles. The energy organelles and the energy transformation unit we are coming up on, I think next week or the week after that, we will actually look into the complexities of these two organelles specifically, the chloroplast and the mitochondria. These amazing little structures are able to transform energy. So in tandem, the chloroplast and mitochondria work to essentially transform the energy of the sun into a type of chemical energy that cells can use. And it's interesting to note that these two organelles are sort of like little organisms in and of themselves. They have their own DNA, ribosomes, and enzymes. And it's interesting that some scientists think at one point in time these were free living creatures and got engulfed by a larger cell. That's called the endosymbiotic theory. It's interesting, but that comes from the fact that these have their own DNA, ribosomes, and enzymes. So they are thought to have at one point been able to survive on their own. Chloroplasts are only found in plant cells and some protists, and they're the site of photosynthesis. And what photosynthesis is, in a nutshell, for this chapter, it is the transformation of solar energy into the chemical energy of sugar or glucose. So again, you should recognize that macromolecule from chapter 3. This was a simple carbohydrate or a simple sugar. Mitochondria are pretty much in everything. Plants, animals, fungi, protists. Okay, do not forget that plants have both. Plants have both of these. Animal cells only have mitochondria though. And these are the site of cellular respiration. And this is where the sugar made from a plant cell is converted into another chemical called ATP. And ATP, and we'll look at the structure of that when we cover cellular respiration, but it is a form of energy the cell can utilize. The cell cannot directly use glucose for energy. It has to be modified and repackaged into this form here, and that is what our mitochondria do. The internal cellular skeleton, all eukaryotic cells have this, and again, it is the internal scaffolding made of protein fibers, and it can aid in movement in some cells, but for the majority of the time, it's just going to be an internal skeleton system. And lastly, the various movement apparati for cells, flagella and cilia. Again, flagella are long and whip-like, so prokaryotes will have both of these. Even human cells have these. So the human sperm cell, for example, has a prominent flagella. And compare that with different types of movement appendages called cilia. And these are smaller hair-like structures that will typically surround the cell, and it will enable the cell to sort of swim and move freely on its own as well. And that's all I have for chapter 4. Again, these are just the main highlights. Make sure you read the chapter a couple of times and take notes from there. And as always, I hope you have a great week, and let me know if you have any questions.